Microbial culture technique. Culture media are used for quality control test of non-sterile raw materials and finished products as well as for microbial contamination. Test in applications such as hygiene monitoring, sterilization process, validation and determination of the effectiveness of preservatives and antimicrobial agents. Culture methods includes streak culture, pour plate culture, liquid culture, lawn culture and straw culture. Streak culture. It is used for the isolation of bacteria in pure culture from clinical specimens. Platinum wire or nichrome wire is used. One loopful of the specimen is transferred onto the surface of a well-dried plate, spread over a small area at the periphery. The inoculum is then distributed thinly over the plate by streaking it with a loop in a series of parallel lines in different segments of the plate. On incubation, separated colonies are obtained over the last series of streak. Pour plate culture. Tubes containing 15 ml of the agar medium are melted and left to cool in a water bath at 45 degrees Celsius to 50 degrees Celsius. Dilution of the inoculum are added in 1 ml of volume to the molten agar mixed well. Contents poured in sterile petri dishes and allowed to set. After incubation, colonies will be seen distributed throughout the depth of the medium. Enumerated using colony counters. Gives an estimated of the viable bacterial count in suspension and is the recommended method for quantitative urine culture. Anaerobic culture method. Anaerobic bacteria differ in their requirement and sensitive to oxygen. Clostridium tetani is a strict anaerobe, grows at an oxygen tension less than 2 mmHg. The method includes production of vacuum, displacement of oxygen with other gases, chemical method, biological method and reduction of medium. First one is the production of vacuum. Incubate the cultures in a vacuum desiccators. Second one is the displacement of oxygen with other gases. Example is candle jar. Displacement of oxygen with nitrogen, helium or carbon dioxide. Third one is chemical method. Alkaline pyrogallo absorbs oxygen. Macintosh flies anaerobic jar consists of a metal jar or glass jar with metal lid which can be clamped airtight. The lid has two tubes, gas inlet and gas outlet. The lid has two terminals connected to electrical supply. Under the lid, small grooved porcelain spool wrapped with a layer of pelletized asbestos. Gas pack. Commercially available disposable envelope contains chemicals which generate hydrogen and carbon dioxide on addition of water. Called catalyst permits combination of hydrogen and oxygen. Indicator is used. Reduce methylene blue, colorless anaerobically, blue color on exposure to oxygen. Biological method. Absorption of oxygen by incubation with anaerobic bacteria, germinating seeds or chopped vegetables. Reduction of oxygen by using reducing agents 1% of glucose, 0.1% of thioglycolate.